So I was running, and I jumped when I was going like this, but stop. The back pushed me, and I fly back right into the pool. I was just supposed to stop, but then I guess I like overshot, and he just pushed me back. I was fucking really hard.
Why were you running?
I was planning to make a really loud noise as I got down. I was going like that, scared him out, but he turned around and he pushed me really hard.
Online thing.
It's the iPad.
I did, I responded. For sure I did.
Do you remember what you said?
What was the question? Was it?
I was supposed to.
I said yes. Yeah, I think I said sure. Whatever your question was.
No, I didn't. I asked what the next possible date was. I gave tomorrow during the day.
Yeah, that's fine.
Yeah, I was like, because he pushed me back so hard. He was like, it's just really funny. I mean, you could have thought it was fun. Or would you like cry and probably push me?
Oh, just go laughing. My friend was one time she was raised on recommended laughing so hard. My friend was in the car and she like went
Okay, everybody right now should have out a piece of paper and they're going to be working on their warm-up right now. Mitosis worksheet that had the definitions of what went on in each phase on the front and the labeled drawings and pictures on the back. The back should look something like that, right? Something. I mean, maybe you were looking at a different image and you labeled it a little bit more, a little bit less, whatever. But at the end of the day, that should have what was been on the back. Correct? Yeah. Can't hear you. I can't hear you. Fine, as long as it's to the bend. So everybody else should have a paper out doing their warm up. Warm up number 13. Yeah. No, you just have to number it 1 through 9. I'm going to call on 3 people. First person's going to give me 1 and 2. Second person's going to give me 3, 4, and 5. Last person's going to give me 6, 7, 8, 9.
I got stuff done.
Can I hear? McKenna, not here? Okay.
Wait.
I swear this thing knows who's absent every single time. Oh, I think we're all just kidding.
The first three?
No, just number 1 and 2.
Interface.
Number 1 is interphase 2, and number 2 is incorrect. Nobody's got number 2 right.
I got it.
What'd you put?
Mitosis.
No, that's incorrect.
He just said it. Oh, that's what I said.
No one has gotten number 2 right.
Cell division.
That's actually closer to the correct answer. It is the M, the letter M. What am I trying to throw?
M phase.
M phase. Now, don't get me wrong, ladies. M does stand for mitosis, but you're assuming that only mitosis occurs in this phase, and that is incorrect. Number 2 is the M phase. Number 1 is interphase. Okay, and that is all of this. M phase is mitosis, which is these 4, and cytokinesis. You see, mitosis is specifically the division of the nucleus, how we copy and divide our DNA. I can divide the nucleus and still not have divided my cell. I would have 2 cells in 2 nuclei in 1 cell. Cytokinesis, cyto meaning cell, kinesis referring to the movement or the pinching in of the cytoplasm, is where we actually split the cell into 2 daughter cells. That's what we call 2 cells that we make from the parent cell. They're called daughter cells. Now, what is 3, 4, 5? Good one. Sammy, 3 is?
Oh, 3. G1.
G1, 4 is?
G2. Synthesis.
So the S phase, which stands for synthesis. And 5 is?
G2.
Huh?
G2.
G2. The G's are like where there's growing. S is synthesis. Synthesis of what? Who, by raise of hand, can tell me what's going on in S phase? Yes. DNA. We are copying our DNA. Remember, common sense would tell me that if I want to make 2 identical copies of a cell, then I also have to copy all of the DNA. So the process in which we're copying all of our DNA, the most important molecules in the cell, happens here. S for synthesis. To synthesize something means to build. What are we synthesizing? DNA. And then in G2, that's where we make sure that we are ready to actually divide. In G2, there's further growth. We make sure we have enough enzymes. We make sure we have enough organelles. The cell's big enough. Right? And as long as all the DNA is copied and attached to itself, we can now enter into what 6, 7, 8, 9 is, which is mitosis. Mitosis has 4 phases. Can you please give me 6, 7, 8, 9? Jordan. PMAT, exactly. Prophase, metaphase, anaphase, telophase. In prophase, right, we move centrioles to opposite ends of the cell. We build spindle fibers and eventually attach them to the chromosomes. The chromatin is condensing into chromosomes, right? And then the nuclear membrane is disappearing. I took 7 cell phones yesterday, guys. Yeah, I'm on a mission. Okay, so there's a lot that goes on in prophase. And here's something important that's going to relate later on when you're answering questions in today's lab. Prophase has a lot of stuff going on. So that means it should take a lot of time or a little bit of time. A lot of time. So if you were to time mitosis from the start to the finish, which phase do you think the cell is going to be in the longest?
Interphase.
No, just mitosis. Just mitosis. Prophase. Now, if you were to time the whole cell cycle, how long the entire cell was in, then interphase is definitely the longest. Metaphase lines up in the middle, anaphase pulls apart, those are really quick. Telophase, everything goes back to normal. The DNA uncoils itself again. Right? Because remember, DNA is like this when it's a chromosome. Right? It's uncoiled like this in telophase at the end, exactly. And in interphase. So interphase, it's chromatin. Right? It's all this stuff, unwound. This is chromatin in interphase. Except it's wrapped around these tiny little proteins. So imagine having tiny little marbles and these are wrapped around it. But it's still long, okay? Now then I copy all of this. I make 2 of these strings. That happens during S phase. I still haven't coiled it up and form a chromosome, okay? So if I made a copy, let's say I do this. This would be the best way to do this. Let's say this is... Okay. So here's my chromosome, right? Here's chromosome number 1, and I make a copy. Yeah, okay, I made a copy. See how it's magic? Let's look at this copy. Okay. Okay, I made a copy, right? These are sister chromatids. Okay. This is dad's number 1. This is mom's number 1. I have 46 of these chromosomes. Right? Dad gave me 23, and here's a copy of dad's. Now moms are in the nucleus too. Now during prophase, right? I got to coil it up. And form what? Chromosome. And then I attach these in the middle. And that's my little X. Attach these in the middle. Got it? Dad has 23 of these. Mom has 23 of these. Number 1 would match. Match, match, match. Those are called homologous
Pairs.
except for that last pair. The first 22 of humans is called autosomes. That last pair, the sex chromosomes, they don't always match because dad has a Y and mom gave him and an X, and they don't match. They're not the same genes. Okay, that being said, that's done. What I need to, yes?
What if a man has XYX, like a mutation?
2 X's and a Y?
Yeah.
He has a condition that's called Klinefelter syndrome. And he is a male because when it comes to sex, the presence or absence of the Y chromosome determines biological sex. So if you have a Y in your cells, you're a man, biologically speaking. And if you don't, if you lack a Y, you are female, biologically speaking. Now, which gender you choose to associate with in society, that's a societal term, that's a sociology term. That you can change your gender, but I cannot remove or change which chromosomes I have in my cells after I'm born. So if I have 1 X and nothing else, right, I have Turner syndrome. Okay? There's another condition. Are you male or female?
Female.
Female, right? Now there is a lot of things affected, not just reproductive organs. Hearts, the skin is affected, your heart is affected. A lot of things are affected, hormone levels. But also you'd be sterile as a female. Same thing with Klinefelter syndrome. Because you're messing with things that that deal with sexuality or sexual reproduction, that individual is also sterile. Okay? But I have a whole PowerPoint all about that. Let's save that when we deal with genetics. But last quick question.
What is it possible for somebody to have like a Y and a Y and no X?
If you had 2 Ys, right, and no X, because the X is so important for the vital functioning of the organism, if a sperm that had 2 Y, which is possible, fertilized an egg that had no X's because of this thing called chromosomal non-disjunction, that fertilized egg would abort. It would miscarry. It would not develop into a fetus or embryo. Got it? So you'd never know. Is it possible to occur? Yeah. Today we're going to talk about cancer. Today's objective is to talk about what happens when the cell cycle goes awry. You should have already gone over, we've already talked about all the steps of the cell cycle. And specifically, when the cell's dividing, what goes on in mitosis to coordinate that. Go out fast. So, any extra what I'm saying? Oh, yeah, that's it. All right. So when the cell grows and divides and grows and divides and grows and divides, Causing the cell to divide, we're going to refer to as like a gas pedal. Go, 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 divide. But eventually we have to hit the brakes and we have to stop. Cells will grow, 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 and then they have to stop. The regulation, what regulates, what controls this, okay, is important. Because if it's broken, we have cancer. There are things that turn on and turn off, or regulate or control the cell cycle. There are external factors, and there are internal factors. Things that go on the outside of a cell that cause a cell to divide or stop dividing, and things that are going on the inside of a cell that tell a cell to start or stop dividing. Let's say I have a petri dish, and I place one cell in the middle, and I trigger it to divide by adding some sort of chemical or whatever. It starts to divide, 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 divide. 2, 4, 8, 16. Fills up the whole petri dish. When those cells bump into each other and to other things, they will stop growing. That is an external factor, a physical external factor. Just like when you cut yourself, this is something on the outside of the cell that will trigger it to divide. When you cut yourself, those skin cells will start to divide and fill in the gap. And when they run into each other, they stop. You can imagine what would happen if when they bumped into each other, they didn't stop. They kept on growing. Every little nick, every little cut would result in giant tumors. Now luckily, our cells have external factors, things on the outside of them that tell them to stop, like bumping into another one. There are also external factors that tell them to start, like growth factors or growth hormones. Chemicals that are produced outside of the cell that when they bond to the surface of the cell, proteins on the outside of the cell, will tell that cell to divide. Now when you guys went through puberty, you hit a growth spurt, right? At some point in your life, you just grew a lot, yeah? Now there was a hormone that's released that triggers growth. It's actually released in your brain from your pituitary gland. It's called a growth hormone. It gets dumped in your blood, and then that hormone bonds to all these other cells, telling them to do what? To divide. To divide. Cells don't get larger. They get more in number, right? That's how we get longer, we get larger. So these are things on the outside of a cell that can cause it to divide. What's going on the inside of a cell? So what's going on when that hormone touches the outside of the cell? Let's zoom in on the inside and see what's going on. How does that cell now tell itself to divide? The cell has two types of chemicals, right? Because the outside triggers internal factors. There's molecules called kinases. These are enzymes. And other molecules called cyclins. Now the word cycle refers to cycling, or cyclin refers to cycle. These are molecules that control the cell's cycle. And when a cell's about to divide, it makes more and more and more and more cycle. So let's look at this graph and see if you can make sense of this graph, because this graph might show up on a test. Up here you have these arrows. It shows mitosis, interphase, mitosis, interphase, mitosis. So you got a cell that's in interphase, divides in mitosis. Then it stops. Each one of those two cells will go individually into interphase. They go into interphase, and then they both divide into mitosis. Now you have 4 cells, right? Then they go in. What's going on inside of each one of those cells during interphase? Well, this molecule cyclin starts to build up. When cyclin bonds to this molecule kinase, it produces a chemical called a mitosis promoting factor, or something that will promote or turn on mitosis. And so when cyclin gets high enough in the cell, in terms of its concentration, it will turn on the activity of the mitosis promoting factor. And so when these two things are really, really high levels in the cell divides. We're in mitosis. But here's the key. What happens after a cell divides to the level of cyclin inside of a cell? It goes down. So something must have happened to that chemical. The chemical must have broken itself down. We call that degradation. It gets degraded. So when there's low levels of cyclin, the cell stops. So it's this amount of chemical inside of the cell that tells the cell to divide and stop dividing. I can actually take a needle and suck it up the cytoplasm from a cell that has a lot of cyclin in it and inject that cyclin that's mixed in the cytoplasm into a cell that's not doing anything and force it to divide. So a cell divides when it has a lot of these molecules in it. What would happen if the cyclin never broke down and it stayed high? What would happen if the cyclin never went down and it stayed at high levels inside of the cell? Drawer.
It would keep reproducing or dividing.
Bloop, 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 and it wouldn't stop. You know what we call that? Cancer. That's cancer. Cancer is where you cannot regulate the cell cycle. It doesn't know how to stop and start normally. You have flying through the stop signs. And the main stop sign is right here, in between G1, see that right there, and G2. I'm sorry, and S. This is a chilling phase of the interphase. You're just growing, doing your everyday activities. This is where you copy the DNA. Why would you want to copy the DNA? Because you've decided that you need to make a copy of yourself. So a cell never enters into S unless it's about to divide. And once it flies through that stop sign, it's going to divide and then go back into G1. So what are some of the things that tell a cell to go from G1 into S phase? A lot of things. It's complicated, right? It's from a college textbook. Of course you don't have to know all that stuff, right? I show you that image to show you that there are many, many factors that will trigger a cell to not only divide, but also to stop dividing. And if we screw with the DNA that controls a lot of these genes, then this mechanism breaks down. And you can't hit the brakes right before G1 and S phase. It's the DNA that codes for the proteins that regulates all these. The proteins that control cyclin and kinase. So let's say you have a cell that's DNA has been damaged, and it's going to be cancerous. You damage the DNA. A cell has this unique self-destruct button. It has this process where it just kills itself. It falls on its own sword. And it destroys itself from the inside out. That process is called apoptosis, programmed cell death. Why would a cell want to kill itself? Well, because its DNA is damaged, and if it doesn't kill itself, it's going to continue to divide and divide and divide, and possibly become cancerous if the DNA that's damaged happens to be on a gene that controls the cell cycle. Another cell that goes through apoptosis is the cells of the tail of a tadpole. Now, tadpole tails don't just fall off the tadpole; they shrink, right? As it grows up. How do you shrink if you're a living organism? Well, the cells actually destroy themselves. In humans, when we're in our embryo, if you've ever looked at a little tiny baby, or when it's like 9 weeks inside of the mother's womb, their fingers are like webs, right? They don't have individual fingers, it's all skins, right? Little bumps. Well, how do we separate the fingers as we grow up? Those cells on the middle will actually destroy themselves, and it separates. It's like cutting, right? Apoptosis. Sometimes this process doesn't function correctly in the fingers or toes, right? And you have people who are born with toes and fingers that are stuck together because those cells didn't naturally go through apoptosis, okay? But it's really a good mechanism in killing cells before they become cancer. So if apoptosis doesn't work, and we've damaged our DNA, and we've broken our brake pedal to stop and regulate cell cycle, we have cancer. We have uncontrolled cell growth, and the cells will divide and divide and divide and divide in this petri dish until we have a big clump of cancerous cells called what? What's a big clump of cells called? It's not a tumor.
I saw that.
Right. Isn't that not the greatest movie of all time?
What?
I will show you a clip.
The kindergarten club?
Yes, thank
I
you.
love that movie. It was on the other day.
You need to get out right now. That is one of his best movies of all time.
Yeah.
Detective John Kimball. Who's that? Get down! Get in the chopper! Who's your daddy? What did you do? Are these your lunches? Stop eating people's lunches! Stop it! No? It's fantastic.
Who doesn't remember that? It's so cute. Like the
You didn't like that movie?
Like I love it.
You're not a good person.
You
Something you may not even have a soul if you don't like Kindergarten Cop. It's his best movie, hands down.
No.
Yes.
Yeah.
Yeah.
What would his best movie be?
Terminator.
Which one? The first one or the second one?
First one.
The first one?
The first one was really good.
That was good. Really good movie. I like the scene in The Predator where he meets his friend Dylan, and then like they do this thing like this, and they're all like flexing their arm. They're like, oh, right? He's like, what's the matter, Dylan? See, I have you pushing too many pencils. You don't remember that scene? Yeah. Yeah. No. Okay. Well, YouTube it. You're gonna see the movie from Kindergarten Cop in a minute. There's two types of tumors. There's two types of tumors. Benign, and what's the second one?
Oh, cancer.
Malignant. Malignant. Comes from the Latin root, B-E-N, ben, which means good, right? What's the Spanish, French, Portuguese word for good?
Bueno.
Yeah. There's a lot of words that have B-E-N in front of them, like benefit, that mean good. Benefactor, benevolent, benign. This is good.
Banana.
Not B-A-N. But let's say this is cells in the top of your skin. We have a tumor. And they've grown. Stop. They stop growing and they don't spread. That's a benign tumor. How do I know if it's benign? Well, I can take what's called a biopsy. I take a little tiny needle and I pinch off some cells. And I can look at those cells. I can look at them in a petri dish and see how they grow and look at their DNA and run tests as an oncologist to determine whether or not that tumor is going to continue to grow and be harmful to you, or if it's done growing and it's benign. Now, if it's not benign, if it's malignant, then it's going to enter into stage 2 or stage 3 or stage 4 of cancer. Okay, these are the different stages of cancer, right? Now, once it starts to grow and break into different tissues, you've gotten into stage 2. When it flakes off into either the blood or the lymphatic system, when one cell of this clumped tumor breaks off and frees itself, now it's jumped on like a hitchhiker onto the freeway system, which is your blood vessels, and now it can travel wherever. So it travels throughout the blood vessels or the lymphatic system. And then now it leaves one of those cells, exits the capillary, and then wedges its way into some other tissue in a different location. So you started off with a skin cancer, let's say in the back of your neck or something, and you ended up with a tumor now growing in your brain, or growing in your heart, or growing in your lungs. When we started off with one tumor and it is spread to have secondary tumors, that process is called metastasis. The cancer is said to have metastasized. When cancer metastasizes, right, when it goes through metastasis, it's spread. Cancer is only dangerous if it spreads into different organs and starts growing there and damaging those organs. Having a big tumor on my skin is not going to kill me. Having that tumor break off and then grow in my brain and in my lungs and in my heart to where that clump of cells sucks up all the blood from that organ and starts to affect the function of that organ, that will kill me. A tumor in my heart, I die of heart failure. A tumor in my brain, I die of being slipped in a coma, or wherever it's growing, it damages the brain's function somehow. If I have a tumor in my lungs, okay, I'm going to have problems getting air into my blood, or oxygen. Yes?
Okay, this might have nothing to do with it, but like, you know, like in like The Fault in Our Stars, where she like had like some cancer, and then like she got rid of it, but like her lungs kept like filling with like water after. Does that at all have?
Okay, I only saw half the movie and then I fell asleep.
How? But basically, like she like her lungs like fill with like
I know
water.